by earlier humans, but it's equally likely that the original population might have been dispersed, or plant might have survived on the floating rafts on the sea. The species showed here is, I think, Rhynchoglas publicum. It's widely distributed in Asia, ranging from southern India, New Guinea, as well as to southern part of China. Middletonia, this is a recently described genus based on molecular phylogenetics study. It's named after Professor Middleton at Singapore Botanical Garden. We have one species in India, rest of them are mostly in Thailand and adjacent areas. So I'll come to the genus that I study, Didymocarpus. It's a genus which was described by a botanist called Nathaniel Wallich in 1819 based on specimens collected by his local assistants in Nepal. He was appointed as kind of superintendent of the Kolkata Botanical Gardens for some time. During his appointment, he prepared an extensive catalog of plants from Indian subcontinent. And this is one of the genera which was included in that. Genus Didymocarpus was loosely defined by few floral characters initially. Thus, it became kind of a mixed bag of species from wide geographic area. So there were species described from Africa, Asia, as well as Australia, which had varying morphologies. Since the establishment of genus, there have been at least 450 names proposed in this group. And the recent revision has considerably redefined the genus. They separated most of the species from Didymocarpus and moved into a new resurrected genus called Henkelia. And some of the African ones were transferred to Streptocarpus. So currently it has around 110 species. Most of them are distributed, sorry, most of them are distributed in China, India, and Thailand. They are lithophytes. Most of them are epiphytic or terrestrial. Most of them are lithophytes. There are some epiphyte and terrestrial plants. It can be easily distinguished from its related genera from a few characters. Didymocarpus has kind of seasonal growth pattern, whereas Henkelia has throughout the season, you can see them. In flowers of Didymocarpus also has kind of capitate stigma, whereas Henkelia and its allies have what they call as chiritoid stigma or bifid stigma. Another important character which distinguishes Didymocarpus from Henkelia is the presence of something called enantiostyle. It's a kind of floral polymorphism where the style deflects to the left or right side of the axis, and is hypothesized to be evolutionary adaptation to increase the cross-pollination. Traditionally, botanists have identified at least two distinct sections, section LRT, and which is most of the Malaysian taxa, characterized by relatively tall stems and lignified axis. There is one more section which some Chinese taxonomists recognize as Heteroboea, consists of some 10 to 12 taxa from Western China, which are nearly ecolescent herbs. And the rest of the species are usually clubbed together as section Didymocarpus. Although these sectional boundaries are not so clear, and there are some genus like Gyrochilus and Allochilus from China, and some taxonomists treat them as part of Didymocarpus. So to bring some taxonomic clarity to the group, we set out to reconstruct a phylogenetic tree and try to understand the species relationship and diversification patterns. So we were wondering whether genus Didymocarpus is monophyletic and how are the species related to each other, given its wide distribution throughout Southeast Asia and yet higher endemism, where and when this genus originated, and are there any morphological characters that distinguishes between different clades or sections of this genus? We try to answer these questions in 4 steps. First, we looked at the taxonomy. We collect the genetic information to build a phylogeny, then to understand the diversification dynamics within genus, and then to have a fresh look at morphological characters in the light of evolutionary understanding. Work of this kind requires us to look at as many as specimens to collect morphological data and tissue for molecular work. Species like Didymocarpus, which have no or very little known economic or aesthetic value, are poorly represented in most of the herbarias. Flora of Assam, which is one of the first comprehensive accounts of plants of Northeast India, very briefly mentions that this genus is of no forest importance. So we also quickly realized that nearly 40% of Indian taxa are not collected in at least last 40 years. And also published diagnoses were kind of incomplete and inadequate. So this project was carried out with RBG, which has a rich collection of Indian plants due to their colonial past, and RBG also has a reputation for work on Gesneriaceae, where people like Dr. B.L. Bird earlier worked on Asian Gesneriaceae and its revision. So we carefully looked at all the specimens, the past collections of Gesneriaceae, especially Didymocarpus in Indian and European herbaria. We also managed to do some 8 months of field work across 3 years in 8 northeastern states of India, and we collected morphological as well as tissues for molecular data. But a note about this kind of habitat in Northeast India, it's kind of relatively unexplored, but recent years it has habitat is rapidly changing. The karst mountains where these Didymocarpus grow is under severe threat, and because of kind of illegal mining and also road construction and such activities. Despite this, we managed to collect some 23 out of 26 taxa that we have in India. This also included some of the interesting collections. For example, these 4 species had not been collected for the past 90 years, and fresh collections allowed us to re-describe them. In addition to that, we were also able to describe some undescribed taxa. For example, this one, which we named as Didymocarpus cynoindicus, was first collected by Kingdon Ward, but that specimen did not have any flowers, so he had not described any. He had not described it as a species. My colleague Preeti collected fresh specimens from the same type locality, and we were able to describe it as a new species. Similarly, another species was Didymocarpus wikifunkiae, named after Vinita's mentor, Wikifang. So this earlier, the species in India was treated as same as whatever is in Thailand, but careful examination of specimen revealed that it requires its own name. For reconstructing the phylogeny, we used the standard 4 standard barcoding markers, and we used the Sanger sequencing to and compared the sequences with whatever we had in GenBank and to build the tree with the parsimony, likelihood, and Bayesian methods. Here is the phylogenetic tree. This is Didymocarpus sensu stricto. It's strict sense. It can be divided into 5 clades. Clade 1 is mostly the Malaysian taxa. Clade 2 has species mostly species which are not traditionally assigned to any of those clades, or in most of these are kind of Thai species. Clade 3 is mostly the taxa from China. Clade 4 has Malaysian as well as some of the Indo-Burmese taxa. Clade 5 was dominated by Indian taxa. So the genus Gyrochilus was sister to some of the Eastern Chinese Didymocarpus, which were acaulescent. We also dated the phylogeny to understand when the genus originated. Unfortunately, there are no known fossils in the group, so we had to rely on the secondary calibration points. We found that the Didymocarpus originated in Miocene at around 18 million years ago. Didymocarpus sensu stricto, if we exclude the Gyrochilus and others, it was originated around 13 million years ago, and it diversified during the late Miocene period. Ancestral range reconstruction reveals that probably it originated in Malay Peninsula and Sumatra region, and it at around like the most recent common ancestor of this Didymocarpus was in Malay Peninsula, and then it further moved northwards and diversified in the Indo-Malayan realm, especially in around the Himalayas. We did the character state reconstruction, so I'll just briefly select a few characters and show them here. Sorry. Most of the taxa from Western China, including those of Gyrochilus, are kind of acaulescent. The ancestral trait probably was acaulescent, and most of the Didymocarpus sensu stricto are caulescent species. Calyx shape is one of the important diagnostic character in this group. Most of the clades have, for example, Indian clade is dominated by campanulate kind of calyx, whereas the Indo-Malayan has mostly the tubular ones. Similarly, calyx tube color, the ancestral Malayan ones were yellow, whereas Gyrochilus and allies are kind of red or orangish ones. That was the yellow was probably the ancestral color in Didymocarpus, and then when it diversified in the north, it mostly turned to most of the species are violet or purple. The same trend is followed by corolla lobe color. Also, the Malayan taxa have kind of yellowish trait, and then as you move to species diversified in the north, it has kind of violet purple colors. And sorry, the ancestral corolla tube of this Didymocarpus sensu stricto was probably personate. I'll just yeah. So just to summarize this, there is still a lot of unexplored diversity of Gesneriaceae in India, and we used Didymocarpus as a model to understand what is going on in this group. Didymocarpus sensu stricto forms a monophyletic group. It mostly originated in Malay Peninsula during mid-Miocene and diversified further in the Indo-Malayan region. Taxa from India did not form a monophyletic group, suggesting possibility of multiple introduction to Indian subcontinent at various times. Northward expansion of Didymocarpus was coupled with floral color change, mainly from yellow to purple or violet. Although clay defining key macromorphological characters were not there, a combination of characters can be used to define these clades in Didymocarpus. Yeah, I thank Vinita, Dr. Michael Moeller and Bhaskar and all the funding agencies. Yeah, I'll be happy to take any questions.
Thanks, Prasanna. Can we give open for questions? Steve.
Yeah, hi, everybody. So I know that I did some field work in original Pradesh a few years ago. We were collecting Didymocarpus pediculatus, and some of the local people were telling us it was used in traditional medicines for renal diseases.
Yes.
And so the question, I have two questions. Are you aware of any new studies investigating the regulation of calcium absorption, thus kidney stone treatment using the species, and are there other ethnopharmacological studies on other Didymocarpus species used in traditional medicine?
There are quite a few studies on the type species, which is Pediculatus, especially which is there in Nepal and the Himalayan region. There are quite a few studies, but I don't know how good are they. It's mostly they use the extract and see if there is any difference. But traditionally, it is yes used in the Indian traditional medicine as well. Even the commercial, some of the medicines, I have seen that they use it, but I really don't know the much about its medicinal value or how effective it is. Apart from that, people also use it for getting colors, especially in Nepal, they use the root stock to get a kind of red color, which they traditionally use in worshipping and all.
Oh.
There's one, is that a question in the chat? Shall we move to Rhuthuparna, Gabe?
I believe so. Wait, there is a question in the chat, yes. And
Okay.
I guess I should say beforehand that we have some visitors from the Gesneriaceae Society National Chapter here with us. The president of the National Chapter region here is Min Bui, and he asks, are there any efforts currently underway to protect, conserve Gesneriaceae of India?
I'm not aware of any targeted effort for Gesneriaceae. We tried collecting them and then bringing here to Bhopal, but we have kind of very dry weather, so it didn't survive here. But there are protected areas in Northeast India where they usually grow. These protected areas are like targeted for, like the entire forest is protected, and thus Gesneriaceae also get some kind of protection. But usually it's kind of neglected as a understory hub. I'm not aware of any targeted efforts so far.
All right, I'll request, thanks Prasanna for the talk. I'll request Ruthu to start her presentation. Meanwhile, of course, I mean, Prasanna is around, so by the end of Ruthu's talk, if you still have questions for Prasanna, please feel free to ask. Ruthu, are you switching?
Yes, Vinita, yes.
So yeah, of course, while she's doing it, I'll just give you a brief sort of introduction to what we're doing. So in my lab, it's called the Tropical Ecology and Evolution Lab. Two of the plant families is a major focus right now. One is, of course, Gesneriaceae, which is what I've been working on for several past years, almost 2 decades now. And the second one is Gesneriaceae, and which of course Prasanna and 2 other students have been invested in, a few more master students in the past, off and on. And also the third plant family that I'm hoping to expand is Commelinaceae, also closely related to the gingers, which is what we're working on. So most of my master students work in the Western Ghats because that's physically kind of closer to where I'm located, while most of my PhD students work in the northeast of India. And so the projects are sort of divided across multiple different types, as in starting with taxonomy and phylogeny and going into the evolution of floral characters or any other sort of interesting vegetative or habit characters, such as epiphytic nature of plants and so on and so forth. So you will see sort of a recurring sort of ideas in most of the projects where we're trying to connect the dots and trying to get overlapping sort of plant families to see the trend in the biogeography of plants from India to Southeast Asia, because that, like what Prasanna said right in the beginning, we know that the Northeast acts as a corridor for plant movement and therefore also shift in characters from what we see in Southeast Asia and what we see in India, and so on and so forth. So with this, I'll ask Ruthu to start her talk.
Yes, Vinita. Is it visible?
Yeah, yes, Ruthu.
Okay. Good afternoon, everyone. I'm Rhuthuparna. So it's a great pleasure and honor to meet you all today to discuss the Gesneriaceae research going on in tree lab. So Prasanna has already explained the taxonomy, phylogeny, and biogeography of Indian Gesneriaceae. So here I'll be talking about Gesneriaceae from an ecological perspective, particularly reproductive ecology. So I'll try to give a glimpse on the diversity of mating strategies exhibited by old world Gesneriaceae and why it is important to study them. So before getting into the type of mating strategies exhibited by the old world Gesneriaceae, I would like to give an introduction on what is a mating strategy and what different type of mating strategies are present. As we know, plants are immobile organisms depend on different pollinating agents for successful transfer of pollen as well as successful mating. So during the course of evolution, plants have evolved multiple diversity in the structure of flowers, inflorescence, and different mechanism for increase the efficiency of pollen transfer, as well as to limit the deleterious consequences associated with selfing. One such major type of mating strategy is the presence of temporal and spatial partitioning of male and female sex organs, that is anther and stigma. The temporal partitioning of sex organ is known as dichogamy. This is a condition where maturation period of male and female sex organs differs. The second type is the spatial partitioning of male and female sex organs in a flower, that is known as hercogamy. So here I'll be talking about the diversity of dichogamy and hercogamy exhibited by different genera in old world Gesneriaceae. So as mentioned before, dichogamy is the temporal separation of maturation time of male and female sex organs in a flower. So it can be either a protandry or a protogyny. In the case of protandry, the male sex organ matures first, followed by the female sex organ. That is the staminate phase, followed by the pistillate phase. So on the other hand, protogyny, the female sex organ matures first, followed by the male sex organ. So two genera from the family Gesneriaceae exhibit protandry and protogyny. There are more than two genera. So here I'm talking about two. Protandry is exhibited by Escinanthus and protogyny is exhibited by a genera called Oriocaris. So as you can, sorry, I'll use the point. So as you can see here, the structure here is the male sex organ and this red structure here is the female sex organ stigma. So in the case of Escinanthus, when the flower opens, It starts with a male phase, that the male phase is mature first and is fully developed and exposed, which is followed by a short period of overlap, as you can see here, where both the male and female sex organs are together, which is followed by a female phase, where the stigma is mature and fully developed and exposed in the later stage of anthesis. So one hypothesis regarding the function of protandry is that it reduces sexual interference between male and female sex organs, and in turn reduce the chances of occurrence of selfing. So there are few previous observations made on protandry, which suggest that at the beginning of flowering season, you can see a male-dominated sex ratio. That is, there are more male flowers in the population than female flowers. And towards the end of flowering season, the population have a female-dominated sex ratio. That is, there are more female flowers than male flowers. So this observation led to the hypothesis that at the beginning of flowering season, since there are more female flowers compared to there are more male flowers compared to female flowers, the female flowers are kind of in a sea of pollen. So all of the female flowers are receiving pollen. So the female fitness will be higher during the beginning of flowering season, and towards the end of flowering season, there will be more female flowers compared to male flowers. So for male flowers, there are enough number of mates to sire seeds. So the male fitness will be higher towards the end of flowering season. So from Tree Lab, my colleague Sanika Gore is trying to understand the functional aspect of protandry in the species Escinanthus parviflorus. So she is trying to understand how the sex ratio is varying throughout the flowering season, and how this shift in sex ratio is affecting the male and female fitness. So we have completed our first fieldwork in the northeast of India in the past 3 months. So we are still analyzing data, and these are few preliminary observations we made in the field, that Escinanthus parviflorus, the longevity is observed from 5 to 13 plus days, and the transition from male to female phase occurs between 3rd to 7th day of anthesis. So on the right side, I have given a few images of floral visitors we have observed during this time. So the floral visitors of Escinanthus, even though the classical pollination syndrome suggests bird pollination given the red color, narrow tube, and presence of nectar as a reward suggest bird pollination syndrome, we have observed a wide variety of pollinators. Even though the visitation rate is low, we have observed beetles, bumblebees, and butterflies to sunbirds. So the contrasting dichogamy present in Oreocaris is known as protogyny, where the female phase, the female sex organ matures first, followed by the male sex organ. So the type of protogyny exhibited by Oreocaris acaulis is known as the pre-anthetic protogyny, where the mature stigma is exposed before the flower is open, and mature anther developed during the later stage of anthesis. So these are the two contrasting type of dichogamy present in the family. So the next type of mating strategy is hercogamy. So hercogamy is a condition where anther and stigma are spatially separated to avoid self-pollen transfer and selfing. So there are mainly 3 types of spatial partitioning, as you can see here: approach hercogamy, reverse hercogamy, and different type of stylar polymorphism. So in the case of approach hercogamy, it is a condition where the stigma is placed beyond anther. So here you can see there is a clear spatial separation between stigma and anther. On the other hand, reverse ergogamy, where the opposite condition exists, like the anther is positioned beyond stigma. The third class of hercogamy is known as stylar polymorphism. So a general definition for stylar polymorphism is a population having more than one type of floral morph, which differs in the height or the orientation of stigma. So here there are three different kind of stylar polymorphism. The first one is heterostyly, which include distyly and tristyly. The second is stigma height dimorphism, and the third one is enantiostyly. In the case of heterostyly, which include tristyly and distyly, is polymorphism in which populations are composed of morph that differs in the height of stigma and anther. So there is a reciprocal arrangement of stigma and anther in two different floral morphs. And in the case of stigma height dimorphism, as you can see here, the length of anther remains the same, but the length of stigma differs in the two type of morphs. Enantiostyly, on the other hand, is different from the other two type of stylar polymorphism. Here, the morphs are generated because of the deviation of stigma from the main floral axis. So I'll discuss more about enantiostyly later in the talk. So in Old World Gesneriaceae, so far three type of hercogamy have been reported. So the first one is, as I discussed before, is approach hercogamy. So there are many genera in Old World Gesneriaceae where approach hercogamy was reported. I have only added three of them here. The first one is Middletonia, second is Hengelia, and the third is Uriocaris. So here, as you can see, this is stigma and this is anther, and there is a spatial separation between stigma and anther here. The second type of hercogamy is heterostyly. So as I mentioned before, there are two type of morph present in a population, long-styled morph and short-styled morph. In the case of long-styled morph, the stigma is placed beyond anther, and in the case of short-styled morph, as you can see here, the anther is placed beyond stigma. So there is a reciprocal placement of anther and stigma in two different morph in a population. So for enantiostyly, it is really different from the other two stylar polymorphism. So here it's either the stigma alone or the stigma and anther are deflected to the right side, generating an R morph, or to the left side, generating a L morph from the main floral axis. So here, as you can see, this is the stigma and this is anther. And here the stigma and anther both are deflected towards the, deflected from the main floral axis. Here the stigma is deflected to the left side, hence generating an L morph. Here it is deflected towards the right side, hence generating an R morph. So there are a few theoretical models on the function of enantiostyly, like why it was evolved, what is the advantage of enantiostyly, etc. So the theoretical model suggests that they promote pollen transfer between morphs. So the assumed mechanism of this is explained here in this illustration. So this one here is an R morph, and this is here is an L morph. The one represented in red color is a stigma, and one represented in yellow color is an anther. So here the stigma is deflected towards the right side. So this is an R morph, and here it is towards the left side. So this is an L morph. So when the flower is being visited by a pollinator, the pollen here is being selectively deposited on a particular part of the pollinator's body, in case of R morph here. And when the pollinator move towards the opposite morph, that is L morph, it touches the stigma positioned on the left side of the flower and leads to the successful transfer of pollen between morphs. So it was suggested that the occurrence of two floral morph in a population and the selective deposition of pollen on the body of pollinators can promote intermorph cross-pollination. Even though enantiostyle have been discussed often by researchers, there is a severe lack of empirical data on this, like the function and evolution of enantiostyle, especially in a family like Gesneriaceae with many narrow endemic genera, where the question is like why the trait was evolved and what is the advantages it provides. And there is another important notable fact about Gesneriaceae is that there is a possibility of the presence of different classes of enantiostyle in the same family. By different class, I mean there are two different classifications for enantiostyle, that is based on the reciprocity of deflection and based on the morph present on individual. So reciprocity is based on whether the stigma and anther both are deflected from the main floral axis, or if it is the stigma alone is deflected. So here I have given an image of Didymocarpus, Podocarpus. So here, as you can see, both stigma and anther are deflected from the main floral axis. So this is known as reciprocal enantiostyle. And here, as you can see, this is Paraboia rufescens. And here only the stigma is deflected from the main floral axis. This is known as non-reciprocal enantiostyle. On the other hand, there is another classification based on the presence of morph on an individual. So if an individual produces both L and R type of morph on the same plant, it is known as monomorphic enantiostyle. And if an individual produces only one type of morph, that is known as dimorphic enantiostyle. So when it comes to enantiostyle in Gesneriaceae, we are expecting there are three main classes of enantiostyle present. As here you can see, this is Paraboia. This is a non-reciprocal monomorphic enantiostyle. Here the stigma alone is deflected from the main floral axis, and also the plant has both L and R morph on it. So this is a non-reciprocal monomorphic enantiostyle. And the second class here is the reciprocal monomorphic enantiostyle. In both cases, this is Rhabdothamnopsis and this is Didymocarpus. So in both cases, you can see both stigma and anther are deflected from the floral axis, and the plant produces both R and L morph. Looks like a mirror image. The next type of enantiostyle is reciprocal dimorphic enantiostyle. I have put a question mark there because this is not really identified. This was only identified from the images. We need more field research to see whether they are really reciprocal dimorphic enantiostyle. So here also you can see the both stigma and anther are deflected from the main floral axis, and all the flowers produced on a plant have the same kind of floral morph. So the most important thing here is that all this three kind of enantiostyle is present in a single family along with a few other hercogamous conditions. So it is really it would be really interesting to see like what is the evolutionary sequence and how these hercogamous conditions are associated with each other, and what is the ancestral character state for each of these hercogamy. So in Tree Lab right now, we are working on enantiostyle in Didymocarpus. So we are looking into the pollen transfer dynamics, pollination mechanism, variation in morph ratio and how it is affecting the inbreeding rate and overall reproductive fitness of Enantiostylus plants compared to non-Enantiostylus Didymocarpus. Didymocarpus, like Prasanna said, is represented by 110 species. In India, it is represented by 25, out of which 10 are identified as Enantiostylus, and few of the Enantiostylus species are given here. So even though these mating strategies and their functions were discussed by great scientists like Darwin himself and subsequent researchers, there is still a severe lack of studies on these reproductive traits, especially in a family like Gesneriaceae. And it is really important to understand the pollination biology, mating system, and mating strategies in this plant family, as many genera are narrow endemic, and this will help in a better conservation efforts and of the plant as well as the pollinators associated with it. So as Prasanna showed before, like even in the recent field visits, we have observed that we are losing Gesneriaceae habitat to road constructions and mining activities, especially affecting genus like Didymocarpus occur in really pristine habitats like moss-covered rocks. So hence understanding these distribution, pollination biology of these plants will contribute to more conservation efforts. So I would like to thank Vinita, Tree Lab members, and all the funding agencies, forest departments for letting us do the field work. And thank
you. Thank you, Ruthu. I'll open up for questions, please.
Hi, I have a question. Ed here. I'm kind of in the dark. I can put more light on myself. I'm just wondering, the pictures that you showed were, I guess, all bees. There was a cartoon of a bee, and we didn't see any butterflies or beetles. Any comments on that?
Sorry?
Should I repeat it? I'm just wondering, you showed pictures of bees, but not butterflies or beetles. So do you have any comments on the frequencies of the different pollinators and so forth? And do you know any of the identifications of the bees?
For Didymocarpus, so far we have only identified butterflies and bumblebees.
Okay.
So I think bumblebees are the pollinators and butterflies are mainly nectar robbers. But in the case of Eschinanthus, we have seen butterflies, beetles, bumblebees, and birds as well.
Is there any nocturnal pollination?
No, we have set up the camera. We couldn't find any nocturnal pollination.
Okay. What about fragrances?
No fragrance.
Hi, Rhuthuparna.
Yeah.
Yeah, nice talk. Very interesting topic. So I have a question on the pollination biology mating system. Do you know, the system you show promote outcrossing, but do you have any evidence of autogamy, or, you know, the plant have an ability to produce fruit without actual pollination? Do you know the percentage of outcrossing in some of these genera?
Yeah, so in the case of Didymocarpus, we had tried autogamy, and none of those autogamy produced fruits. So they are not able to produce fruits with in the absence of pollinators.
Okay. And what about the selfing? Do you know?
They are self-complicated.
Yeah. So if the flower doesn't produce any pollen, I mean, if the stigma doesn't receive any pollen, is it able to produce fruit anyway? Some percentage?
Sorry?
If a flower, you know, the stigma of a flower doesn't receive any pollen at all, it is still able to produce fruit?
I don't think so, because we haven't seen any fruits in autogamy.
Okay.
Yeah.
Yeah, okay. Thank you.
Yeah, thanks.
So like what Ruthu
on.
sort of described, I mean, there's a little, I mean, I have to say in terms of complexity, this is another sort of a scale, because there's spatial separation and also there's temporal separation. So we're trying to now to sort of, really go into that fine detail of seeing exactly when the separation happens, since it's a multi-day flowers, and how that separation also happens at a population level. Because I strongly believe that there's a population signature that is there in this kind of mating systems, which we're also seeing in other plants, such as some genera in Commelinaceae, where we're seeing that this sex ratio switches is temporarily affected by climatic issues or even the state of the plant. This has been shown in many other Solanaceae also. We've seen this and so it's reasonably common. We're just sort of starting to tease that apart in Gesneriaceae now. Yeah. Sorry, someone had a question. Oh, hi, yeah, Rob.
Do you have any data on seed set relative to the different pollination systems or the different types of pollinators?
We have a seed set from different populations, but like not dependent on different pollinators. So we have pollinator observation data from different population and seed set data also from the same population. So we might be able to see how the seed set changes according to the pollinator visitation rate also.
What's the level of percentage of seed set in these plants? Obviously, they're crossing, but
I have yet to analyze, like do the analysis and seed count and everything. So I just finished the field work and came back to Bhopal.
Typically, it's pretty low in plants that are outcropped like this. But do you have an estimate, or they set a lot of seed or a little seed?
A rough estimate about like how many fruits you've seen in the population, whether you've seen fruits at all. I mean, that will give a good idea.
Regarding fruit set, the one thing is that the pollinator visitation rate is really high, and I have seen almost every bracts producing fruit. So the fruit set rate is really high.
Does it vary how many seed are in the fruits?
Seed, I think there are. I need to look at it.
They just came back from field a few weeks ago, so we're still sifting through the massive data that they've collected. So unfortunately, the fine detail we will not be able to tell right away. So, but we did the crosses, so which is why Ruthu is able to tell you at least the preliminary, observations.
Great. Well, thank you very much. Great talks.
I have a question, but also just to second what Jean said in the chat. Thank you to Ruthu and Prasanna and Vinita for being here in the middle of the night in India. I can hear the chorus of crickets chirping in the background of Vinita's video to attest to the fact that it's 4 o'clock in the morning there. So thank you for waking up so terribly early to be with us. But I was thinking throughout both of these talks from Prasanna's and Ruthu's, are these plants largely lithophytic, growing on rocks? And if so, is the orogeny, like granitic versus limestone areas, do they strongly affect the distributions across the land masses, especially with these strict endemics? Are they restricted to certain altitudes? Does altitude and rock type have a strong effect on distribution?
Yes, at least in Didymocarpus, most of them, can you hear me?
Yes.
Yeah, most of the Didymocarpus are kind of, they grow on rocks, especially kind of karst rocks. So it's kind of a very specialized habitat requirement for them. I think only 2 species are there which are epiphytes. Rest of them are kind of karst-loving plants. Very few are widespread, as I said. The 1 or 2 Rhynchoglossum species, few Corolodiscus species are widespread. They're not so specialized. The rest of them have their own habitat specialization, as well as altitude also.
So is there a good bit of rock climbing involved in doing the field work? Like, are some of these rock crops?
A little bit. So some places we had to climb it and then for collecting, but it's mostly along the streams. You find big boulders of karst, and then you can easily pluck it from there.
Thank you.
Marcos, did you have a question?
Yeah, I was thinking that in the Caribbean we have some species of Gesneriaceae that they only grow in limestone on the forest understory in areas that they're very wet, like around caves, and with dripping water. And yeah, Gabe question make me remember that I was wondering if that also happened, that you have some microhabitats within the same type of substrate. I don't know if that happened also in India, if you can have a species only restricted to some microhabitats.
Yes. Quite a few of them are restricted to just 1 or 2 populations and very close to some waterfall or something, which is very specific to that. Karst habitats are basically limestone habitats. So they have similar requirements here also.
Have you ever done an analysis on the substrate, like the rocks they use based on the phylogeny? Like you did with the flowers?
There is one study from China which looks on the habitat, how the habitat requirement has evolved in that group. Yeah, but that is one of the important diversification factor there.
Okay, thank you.
Michael Riley has a question. It appears that all of these genera are fibrous rooted. They depend on seed dispersion. There are other similar genera that are rhizomatous in Southeast Asia. Is there any relation?
Yeah, we do have both rhizomatous, or it's kind of root stock, and then there are kind of fibrous rooted. Most of the Didymocarpus that I know are kind of rhizomatous ones. I don't know if that dispersal studies are literally not there, so we don't know for any of the species how it is dispersed. It's just a hypothesis for each species, that's it. There are no empirical studies on dispersal. So most likely because dispersal is, at least in Didymocarpus, it's said that dispersal is through rainwater and water flows. So it's restricted distribution is because of that problem.
I mean, I have to say that after many years of doing this, both ecology and evolution in terms of phylogeny, is the definition of a well-studied group is when someone has studied 5 species out of 100 in terms of the ecology, and we call it well-studied. And what we're starting to realize is that, really targeting 80–90% of a particular genus is very important for us to actually understand what is the diversity of mating strategies, ecology, and also morphologies for that matter. So I'm pretty sure that by the time Ruthu finishes her work, I mean, and other students like Sanika, who's doing her master's with us, we may actually end up describing or consolidating some of these mating strategies into newer sort of definitions, because even we are sort of surprised by what we are seeing and what we had read in the literature and what we're seeing, in fact, in the wild. So there's a clear sort of discrepancy in a lot of information. So mating strategies, of course, I mean, it's very difficult to study dispersion, but of course, pollination is a little bit easier, which is why we're starting with pollination, and hopefully we'll get to dispersion through population genetics or using some population genetics type markers to look at how far species move. But that's sort of the eventual goal at some point. And hopefully one of the students will make it to the museum to the PopGen work, if that's a possibility. So I see that John is here. John, do you have any questions? John Bogan and Larry, do you have any questions for us?
Do you have anything about seed dispersal, how the fruits are dispersed?
Ruthu, do you have one answer?
No, we haven't really looked into the dispersal part yet.
Most of them have kind of dry capsules, so most probably they just fall off, and then rainwater carries it to different location. But there are no empirical studies that we are aware
of. Not totally related, but I couldn't help but notice in both of your talks, there's illegal mining happening in the regions where you collect your plants. What are they mining? And are the mine tailings toxic? Is this a major environmental threat in terms of drinking water contamination?
This area has a lot of limestones, which is mined. Limestone mining is there. It's kind of a manual mining. So people just dig out the holes in the hill, just collect the limestones and then sell it. So there is no control over where or how it is done. So it's kind of very risky, plus it's the habitat also degrades that. So every year we go to the field, we'll see that the populations from previous year are totally missing, and there is mining going on, or there will be some new road coming up in that area, and then populations will disappear.
Oh, so this is limestone used for fertilizer, like lime
Yes.
fertilizing.
Yes.
And is this also a major tea growing region? Is this heavily cultivated, or is this too far?
Yes, Assam is known for tea, but Assam plateau is kind of lowland area, and it is surrounded by the highland areas of Indo-Burma and the Himalayan mountain ranges where these Gesneriaceae grow. So it's kind of Assam where tea grows is kind of valley and surrounded by these hills where you see Gesneriaceae and other gingers, which they are very diverse in the surrounding hills.
Wow.
So are these plants easy to cultivate in gardens or greenhouses that you can, for conservation or to do pollination experiments? Is that possible?
We tried for last 3-4 years to bring them to Bhopal and cultivate, but it was not successful. There are only a handful of Gesneriaceae and few Henkelias which are like garden in commercial cultivation. Most of them are their habitat requirements are very specific. So far, we are not able to grow them in our condition here.
Yeah, I think we don't have really greenhouse, or we have only a shade house. There's, of course, the greenhouse kind of does not make much sense in India. But for this, because the climatic conditions where most of the Gesneriaceae grow are very specific and different. We've attempted to grow it in where I'm located in the center of India, but the climatic conditions are too severely different. So most of our studies is done in the field itself. Also, the other part from our ginger sort of experience from gingers, which are technically more hardy rhizomatous, what we're seeing is that in the shade house, we also do see sort of differences in the way the floral morphology looks. So in some sense, I think as far as possible, we will try to look at it in the wild rather than do it in the shade house. Although it is a little difficult, but I think since we're also looking for the sex ratio differences as a factor of population level strategies, therefore it kind of also makes, we may insist on sort of working on this pollination biology in the field rather than in the shade house. Because under higher nutrition conditions, the sex ratios change. So then I think we will not be able to see what we are expecting. In fact, that's part of the problem of many of these mating strategies, because plants were taken from the habitat and then grown in shade houses and studied, and the sex ratio differences are therefore possibly hidden when it's done in a shade house condition, which has high nutrition, enriched soil, and so on and so forth. So we were hopefully going to try to avoid that because of these issues that we may face in sex ratio related questions.
Do you have any cases of bats or other, nocturnal flower visitors? Like white flowers might be visited by moths?
Sanika or Ruthu, do you want to answer?
So, at least for Gesneriaceae, we tried to see if there are nocturnal pollinators, but we were not able to catch any on the cameras that we had. Okay.
So yeah, like Joanna asked about the growing of the plants in botanical gardens. There are botanical gardens in the Northeast. We could, like I said, we could possibly grow them. The only reason we're right now trying to get the plants to Bhopal or to any botanical garden to grow is more so for genomic studies, so that we can get fresh tissue for genomic work and chromosome sort of counting in case of there's any issue of ploidy. But for ecology, like I said, I think the more I study, the more I feel that doing it in the wild is the right way to go, because we will miss patterns otherwise. And when I say patterns, it's like patterns that are under selection for evolutionary trajectories to shift. And so for that, I think studying it in the wild is really critical in their native habitat. Because in the native habitat versus introduced habitat, again, from heliconias, we've seen plants do different things. So I mean, think of it just like Indian, or like, sorry, any race, people are different when they're in their native habitat versus when they're in an introduced habitat. Plants do exactly the same thing. They behave differently. I think we're just starting to see this behavioral differences in native versus introduced habitats. So, yeah. Yes, yes, Jean, I'm at home and Prasanna and Ruthu are in their lab. Good catch. That's what you get for being a faculty. You get to sit at home. Although they had a choice to present it from their hostel rooms, but then I think all three of them decided that they're better off in the lab than in their hostels. So there's a question there for Ruthu and Prasanna. From a cultivar perspective, it is possible that plants grow in an organic substrate that has accumulated on a limestone base. Yes. The answer is yes, if we are asking.
We did try that for some of the Didymocarpus. We brought in the moss along with the rootstock and tried to grow in similar condition here. The leaves will sprout for a few days and then it starts drying. So we were not able to give that micro habitat probably, but ideally that should work.
Okay, if there are no more questions, shall we wind up?
I believe, yeah. I forget the number of seconds you're supposed to count after the last question to know that there's no more questions. But I believe we've reached that point, and you all probably need to get some very well-deserved rest or some very well-deserved breakfast, whichever comes first.
Yeah, it's more like a breakfast. I think it's 6 in the morning.
Yeah. So thank you very, very much for sharing your research with us and the absolutely gorgeous images. These are such fascinating plants, as everyone has said. So yeah, best of luck with all of your research, and stay well and healthy towards the end of the semester. And we will all be in touch, and we'll hopefully be back together next month.
Right. Thank you so much. Yeah, I just wanted to last word. If you have, I know many of you work, of course, the Gesneriaceae Society is here. If you have any more sort of, if you know of people who are doing ecological work and would like to stay in touch with us, please do connect them to us, especially from Southeast Asia, because there of course, the distribution and diversity is high there as well. And as well, if you have any sort of new collections and you think that it will be connected to what we're already doing, we would be more than happy to learn about what you are discovering about the mating strategies and even just floral characters in the new groups that you may be studying in, especially from Asia and Southeast Asia and India. So please feel free to contact us and myself and my students about any questions that are related to this. So thank you, everybody. Nice to see everyone again.
Thank you.
There's
Thank you.
Gabe.
Thank you.
Okay, bye.
Bye.